I'm going to start by asking you guys a question. When you think of nicotine, do you have a positive thought about it or a negative thought about it? I'm going to re-ask this question in a scientific way now. When you think of nicotine in the body, do you think it behaves in the fact that it'll inhibit cell growth or promote cell growth? This was the thesis of my research. Now, initially, before I started my research, I came in with kind of a pre-hypothesis. And I thought nicotine acts as a toxin in the body, so it'll kill cells and thus inhibit cell growth. Now, this would later change. So, now I'm going to list out the preview of what I'm going to be talking about today. And to start out, I'm going to talk about why my hypothesis changed. The methods done in this experiment to obtain the results I got. What my results were. And then finally, what those results mean. And what can I do with these results? So, let's get right into it. I did some some research about nicotine's effect on cells in general, and found that in one research case, nicotine actually stimulates lung cancer cell growth. And this was done by Ying Zhang, Jeffrey D. Ritzenthaler, Jesse Roman, and Shuwei Han. So, now that I knew that previous research had been done, similar to mine, that I was about to do, I had to kind of adjust my pre-hypothesis into the thesis that I had stated before, which was, does nicotine stimulate or inhibit cell growth, Jurkat cell growth? Now, I keep on saying Jurkat cell growth. So what is Jurkats? Well, Jurkats are T cells obtained from a 14-year-old boy that had lymphoma, which is a type of cancer. So these are cancer cells. And they've been immortalized so that they can be reproduced outside the body and can be essentially kept going indefinitely if maintained properly in a sterile environment. So, this was the first part of our methods, maintaining Jurkat cells. So what we do is, you'd have a flask of Jurkat cells, and at the start you'd count, using a hemocytometer under a microscope, the amount alive versus the amount dead, and this would give you a ratio. Now this ratio would then be used to calculate calculate how many cells were alive inside of our flask. So, once the calculations had been done, you then were able to pick out the number of cells that you need for your new flask, because you have to reseed your flask every time, just to keep that sterility going and make sure that your cell line is continuously alive. So, you'd reseed your flask by pipetting the amount of cells you need out of the flask into a new flask, and then adding the rest of media. So these were 10 milliliter flasks. So usually around, I would say about 0.8 mils of cells were continuously added to the new flask, and then the rest was just media for the cells to grow in and thrive in. And the media consists of just simple sugars and stuff for the cells to feed on and grow. So then, after you reseed your flask, we had a maintained cell line. I needed to make dilutions of nicotine, because you can't just test one dilution. That's not going to give you significant results. That's not going to give you conclusive data at all. So I decided to make multiple dilutions of nicotine. So what I did was I did 1/10 dilution, and then from that 1/10 I did a 1/100th dilution of that 1/10 dilution, and I continued this process from taking one dilution and diluting it further all the way down to 1/1 millionth of a dilution. And so so I made these dilutions. Now, the next start was to put these all into a well, into separate wells. So we had wells with 10 wells plates, and so we chose a row. I'd choose a row, and I'd start with a control and just put cells in that. Keep that just nicotine free. And then I put the 1/10 dilution, the 1/100th dilution, and so on, all the way to 1/1 millionth. And I also added a constant amount of cells, the same as the control amount, but in all the dilution wells. So they were all the same concentration at the end. Not the same dilution, but the same concentration of cells. Now, after I had made wells, I did this 3 times, and I took absorbance readings of the 3. And this showed me how many cells were alive, essentially, in all of the plates, in all the dilutions. And that led me to my findings. Now, if we look at the 3 tests here, we can see some patterns. The first one being, there's a steady incline growth of cells in the absorbance readings. From our control, which starts out pretty low, and then our 1/10 dilution, where it jumps up right away, and our 1/100th dilution, where it increases also significantly, but then we also start to notice that cells plateau. The amount of growth isn't as significant as the 1/10 to 1/100th dilution. So this may be significant. There's something there. But the reason why it's plateauing is because we had reached the maximum quota of cells that could thrive in that environment. Essentially, it was just kind of like a maximum populace. So, now I'm going to talk about what I interpreted from these results. The first being, Jurkat cells are stimulated by nicotine. Their cell growth is stimulated. So it was good to kind of do some previous research, because now my hypothesis was better formed for this experiment, instead of just coming in with one test and seeing if it inhibited. I also now see if it stimulated, and it did. So it stimulates cell growth, and I also found maybe an optimal range of concentration of nicotine to use to stimulate cell growth, between the ranges of 1/10 dilution and 1/100th dilution. What it is, I don't know. That might have to be some future research I do, where I limit down the dilutions to maybe a more exact point, so it's not a range anymore. Some more future research I could do is, now that I know that nicotine stimulates cell growth, look at the mechanism behind it. Figure out what the mechanism is. So now let's review what I've talked about. Jurkat cells stimulate cell growth. No, nicotine stimulates cell growth. There's an optimal range I found at which nicotine stimulates cell growth, between 1/10th dilution and 1/100th dilution. Some possible errors could have been pipette errors, so measurement errors, but I don't believe that to be true because of the similarities between the 3. So therefore, it's either I made the same error 3 times, or that wasn't a significant error, and that the strength of my method was actually great. So now, in conclusion, I am now able to comfortably say that nicotine stimulates cell growth in Jurkat cells, so in cancer cells, which is commonly known, but now I have to challenge myself and ask, what is the mechanism behind this? And does it differ between maybe lung cells that the research Ying Zhang, Jeffrey D. Ritzenthal, Jesse Roman, Xu Wei Han did with lungs? Does it differ in Jurkat cells or in lymphoma cancer? So that's something I now have to learn for myself. Thank you.